Okay, welcome everybody. Okay, my name is Andrea Lonick, and I will be your instructor for the next, 14 or 15 weeks in microbiology for the evening class. And I just want to say a little bit about myself. I have been teaching or working at the college since about 2005. I also work at the hospital per diem in hematology and microbiology. So I'm hoping I can bring some clinical information or background to this course as we travel through the journey of microbiology. Before I continue, I wanted to say a couple things. First of all, the microbiology course basics, that will be on Blackboard in the syllabus and the schedule. I really recommend you take a good look at both. The microbiology syllabus has all the information you need about this course, including the assignments, when they are due, and the tests, and also has a grade range in there. If you're wondering sometimes through the mid-semester, what grade am I getting for so far? We have the points in there, and it's in letter grade form. So anyhow, also in the syllabus are the rules and regulations. And I have also included office hours in there, which are your Zoom office hours. I am going to be holding them on Mondays and Wednesdays. Monday from 7 to 8 p.m. and Wednesdays from 12 noon to 1. I will be sending out invitations every week, and if you have any question about the lecture, assignments, or any concerns, please set up a meeting with me. Email me back with your reservation, or just join in. And anyhow, like I said, it is in the syllabus, so give that a check over. Also, the schedule will be in Blackboard, and it only goes up to about the midterm. We've got all the reading assignments and the lecture material included in Blackboard, and some of the links are even on the schedule. Okay. So anyhow, what is microbiology? Microbiology, if you take the word, you can break it into two parts. Biology is a study of life, and micro is small, so it's a study of small life. You need a microscope to see them. And we really weren't able to see them at all before the invention of the microscope. So it is a study of microbes. Another name would be your microorganisms. If you wanted to find out more about microbes or microorganisms, you just go to the internet and Google microorganism, and the first thing you come up with is Wikipedia. A little bit about everything is in Wikipedia. But actually, some of this information is very good. And one of the things is mentioned that most of the microbes are unicellular, so they're single-celled. We do have some microorganisms that are multicellular. And they are diverse, so they come in different shapes, sizes, and forms. There are several categories of microbes, and I'm going to show you that in the next slide. They also live anywhere and everywhere. They have found them in the soil, they found them in the biosphere, also in the hot springs in Colorado, and even in space. Okay. We also know that microbes exist everywhere here on Earth. Okay, you can't touch anything without picking up a microbe. They do live in and on us and in and on other organisms. And one thing about them is that they are pretty much essential. We have found out through the years that they are essential to survival, to our survival. And very, very few of them cause disease. They get a bad rap. So anyhow, let me introduce you to the categories or the groups of microbes. We've got one category that some groups belong to that are cellular. So in other words, they're cells. They're cells. And that would include your fungi. At least there are microscopic phases of the fungi. Protist, which would include your protozoa and algae. Bacteria and archaea. Bacteria and archaea are both prokaryotes. Okay, they are prokaryotes. In the acellular group, so these guys aren't cells, not what we defined as cells. We've got viruses. We also have variations of viruses, smaller in size than a virus, which are your viroids, satellites, and prions. So again, these are classified as microbes, but they are acellular. Now, like I had said a few minutes ago, most microbes, at least 99% of them, are essential to life. We need them to survive. They are found everywhere in all kinds of ecosystems, and they do not, this is 99%, do not cause disease. There may be a few that cause disease, maybe less than 1%, and then a very, very, very rare amount, maybe less than 1/100%, that do cause fatal diseases. And they are horrific. And an example for this less than 1/100% microbe would be your Ebola virus. Now, before I go on, I want to give a little background on this systematics. So what does that mean? Well, we've got to give some credit to the classifying and organizing of these groups. We have many, many types of living organisms, from the microscopic to the macroscopic. And in order to work with them, in order to identify them, they have to have names and be classified. So we give credit to the father of taxonomy, Carolus Linnaeus, for doing this, okay, for initially starting this. So he was able to classify life into groups. They're called taxa or taxon. And also with naming. Naming of these organisms was just as important. So the system we use is called binomial nomenclature. It's actually just two names. It's known as their scientific name. And this is a function of taxonomy. And how we name these organisms is that they use the genus and the species names. So for example, Staphylococcus aureus, a bacteria, is given the name because the first part of the name is Staphylococcus, which is the genus. And you'll notice that it starts with a capital letter, capital S. And the second part of its scientific name is aureus, which begins with a lowercase a. So this is the way we name them and present them, their names, in text. Phylogeny, which is another system, uses or incorporates evolutionary relationships between or among organisms, and it's based on their DNA sequencing. So in this case, the thing that is different in phylogeny from taxonomy is that we are depicting the relationship evolutionarily among organisms. What came before something else in the scheme of evolution? So we've got the timeline here. of the tree of life, of the kingdoms, and where the kingdoms appeared in the evolution of organisms. So first of all, let's give credit to Carolus Linnaeus again, the father of taxonomy. He also proposed that there were two kingdoms, the animal kingdom and plants, in about mid-1700s. About 100 years later, Ernst Haeckel, proposed that there were two additional kingdoms, Monera, which includes bacteria, and also the protist. And then in 1969, Robert Whittaker proposed adding fungi as the fifth kingdom. So this is how the tree of life developed, utilizing the kingdoms. In 1977, though, there was someone else that came along, Carl Woese, and he noted that everything has rRNA. Everything living does have rRNA, a type of ribosomal RNA. And he proposed that based on the type of ribosomal RNA they have, they can be grouped into even bigger, larger groups above the kingdoms, and these were known as domains. So we had the Eukarya domain, the bacteria domain, and now we have a new guy on the scene, the Archaea, which are in the pink balloon down here. He discovered this group, okay, this domain, and the Archaea are similar in some respects to bacteria, but they actually have more characteristics that are closer related to Eukarya. So we give Carl Woese credit for the domain, proposing domains, and also for the discovery of the Archaea. So let's put it all together. This is the overview of the tree of life. And this is actually a phylogenetic tree because we can see the evolution of organisms here. We started out at the moment of life with a universal ancestor. It was a type of very, very primitive cell. And at some point, life had started and it split. The first on the scene would be your bacteria, the domain bacteria. Next, after the split, we'll see that the domain Archaea developed. And finally, Eukarya, way out here on the outer branch. And there we are. We're the babies on the block, animals, kingdom animals. The thing to note, again, I said, is bacteria and Archaea are both prokaryotes. So they don't have a nucleus and cell organelles typical in the Eukarya domain. Okay. And this universal tree is there are always genera and new species added to it as we discover more organisms. The viruses are not included in this because viruses, they are acellular and not considered by many as living. So there is a type of method of grouping them that they do not belong in this tree of life. So before I go on, I want to mention a little bit of the history of microbiology. Way back, even before the discovery of the microscope, humans had known in ancient times that there was such a thing as disease and illness, and that some diseases can cause death. So a disease has always been among us, and it's always been a problem. Back in ancient times, they believed there was some kind of component in the air that caused illness, and that it could spread from one person to the other. They would call it bad air, the miasma theory. But then with the invention of the microscope, and specifically I'm talking about the microscope by Van Leeuwenhoek up here around 1673, he was able to observe live microbes, living microorganisms. And one of the reasons for this is that he developed a high power magnifying lens. It probably magnified about 300 times or more. And that was really a big magnification for that time. So he was able to see things that were living. And one example was that he was able to get a little scraping from his teeth. And it was a handheld microscope. He'd put that scraping on the microscope and then hold it up to the light. He was able to see living things moving around in there, probably some bacteria. I don't know, maybe some protozoa, but he called them animalcules because they could move. So from there, we were able to, as time went on, advance the microscope and discover the world of microbes. And this was a time of great growth, great discoveries, inventions, and even some vaccines. So this is called the Golden Age of Microbiology from about 1857 to about 1920s. And there are two men that are really important during this time. They're called the Golden Men of the Golden Age of Microbiology, and they were Louis Pasteur and Robert Koch. The reason that we were able to see microbes, of course, was because of the advancement of the microscope. But the reason there was an interest in microbiology was because of the fact of disease and illness. Not only disease and illness, but epidemics. The spreading of disease within a population. There was high mortality with some of these, such as cholera and typhoid. So there was a lot of support from the public to research these epidemics and to control these diseases. So this is why it became such a standard, such a priority in the 1800s. So anyhow, and even today, some of the medicine and pharmaceutical industry are rooted from the knowledge and the techniques that were acquired during the golden age of microbiology. So I want to talk about two of those golden men that I mentioned. The first one is Louis Pasteur. And he, as you can see with each one of these bullets, these are just some of his accomplishments. He was determined that fermentation was a chemical process done by living organisms. It didn't just happen. Winemakers knew that fruit juice and grains turned into wine and beer. They didn't know why or how. And Pasteur determined there were microbes doing that. And the microbes just don't magically appear. They actually may be in the air or contaminate the fruit juice or foods and spoil them. What the winemakers had noticed that some of their fruit juice became wine while some of it became acetic acid or vinegar. And they didn't know why it was spoiling. And what Pasteur had determined is that there was a certain type of microbe, the yeast, when that was only mixed with the fruit juice, it would produce alcohol. Whereas if there was another microorganism, let's say a bacteria, through the process of fermentation, it would produce an acidic product or vinegar. So finding this out really made many winemakers happy. And with this, he patented a process known as pasteurization. So, to make it simple, what would happen is Pasteur would have the solution, in this case maybe wine, heated up and then cooled. So it would kill any kind of contaminant, any kind of microbes that weren't supposed to be. It would kill them, be there. It would kill them. He would then add the yeast in this case, and after it cooled a bit, and then you would get wine as your product. So he patented pasteurization. Another thing Pasteur did was he proposed the germ theory of disease. Some of the manufacturers that made silk were having problems with the silkworms producing silk. He found out there was a microbe that they were infected with, and he identified that and found a way to eradicate them so that the silkworms would be able to, or started to produce silk again. Also, Pasteur discovered that weakened microbes that cause disease, when injected into an animal and even a young boy bitten by a rabid dog, would cure them or save them from certain death. Also, all of these accomplishments of Pasteur really awakened the public, or really turned on the public interest into discovering cures to diseases, and these cures would help in medicine. Many people started donating to the cause, and we had the first college, the Institut Pasteur, opened in 1888. Thanks to these donations, and there was the first course of microbiology, now a science, offered at that college. Okay, the second individual is Robert Koch. He really did pay attention or believe in the research that Pasteur was doing on the theories of germs and disease, but he went even a step further and developed an experimental model or 4 steps that will aid in determining whether or not a specific microbe caused a specific disease. And that is known as Koch's postulates. Also, there were people that worked with him that invented some things that are very practical and taken for granted today in the lab, but that really promoted microbiology. And one of them was a petri dish, the little dish in which you have seen. They were glass back then, they're plastic now, where you could grow the microbes. But you also needed the food, the agar. And Walter Hess, and especially his wife, shout out here to Angelina, formulated a medium, an agar, that would allow bacteria to grow. And what she added was actually, it was kind of a thickener. She suggested a thickener that would, when added to the media, would make it solid, so that you would have the microbes growing on top of it, instead of mixing in with a liquid media and not being able to isolate them. Other things, so because of that, they were able to grow microbes on a pure culture, in culture. He also developed certain staining methods that helped in identifying bacteria causing diseases such as tuberculosis. And because of his research with tuberculosis, he was granted the Nobel Prize in Physiology in 1905. So this is a picture or a diagram of the postulates, of Koch's postulates. And postulate number 1 is you take a sick animal. In this case, it is, we'll use the bacteria Anthrax bacilli, which causes anthrax. And anthrax was pretty common among sheep initially. So postulate number 1, the organism or the same organisms are present in every case of the disease. So they would know with certain signs or symptoms displayed by the sheep, they would know, or they would pretty much have a good guess that the same microorganism was affecting those sheep. So what they would do is take the tissue from the dead animal, because it was very fatal, and for postulate 2, that microorganism was isolated from the tissue and grown in a pure culture, because now they had the means by which to grow these, the means and the media in which to grow these microorganisms. That would lead to postulate 3. So once you have a pure culture, what they would do then is take some of those microbes from the pure culture and inject it into a healthy animal. That would be your postulate number 3, which they used mice here. They show a mouse. The mouse would get the disease, show the signs and symptoms, and die with the anthrax. Anyhow, and then from that dead animal, they would again take and isolate some of the microorganism from it and grow it in pure culture, and then identify it as the same microorganism that was initially present in the sheep, in the first animal. So this was a very, very tidy, step-by-step group of rules. But it has limits, and today we know that disease presentation may be different or vary among patients. So postulate 1 doesn't always hold true. Also, infections don't always lead to disease. So, for example, in poliovirus, a person may be infected with the virus, but it only causes disease in about 1% of those infected. Another limitation is that most microbes cannot be grown in culture. So postulates 2 and 4 cannot be fulfilled, cannot stick by those. And then finally, postulate 3, inoculation of a pure culture into a healthy, susceptible organism, and they reproduce the disease. That is unethical if the disease only infects humans. So there are some limitations to these rules and exceptions. I just wanted you to be aware of that. So now coming up to the age of the microbiome. And there are 2 individuals here that we give credit to. Since the 21st century, so the early 2000s, we have realized that we are not alone, that there are microorganisms that live in every ecosystem, that live, like I said, in and on us even. And we can give credit, first of all, to the man on the left there. It is Rene Dubois. He studied ecosystems. He's also an environmentalist. And he basically said that microbes live in communities, live in systems. They do not usually perform or exist in isolated environments. So, in other words, what we can do now is we can grow some microbes, but we take them out of their natural ecosystem. We would feed them, we give them nutrients, we give them shelter in incubators, good environmental conditions. They actually become sort of a tamed pet. It would be like bringing a raccoon in from the wild. You find a baby raccoon, you bring it in, you feed it. It's got a It's got a nice little pillow to sit on and sleep and protect it. It will not learn how to survive out in the wilderness. In fact, in many ways, it's they try to discourage you taking in wild animals because, as you know, once they are domesticated, they cannot be released in the wild because there are certain things, behaviors that change. And so Rene Dubois was saying that. Another gentleman is Joshua Lederberg, and he termed the word microbiome. And it basically, the definition of microbiome is right here in the words in the little essay here. That is basically a community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. So we have microbiomes in our gut, on the surface of our skin, and even in other areas of our body that normally can inhabit that area. So anyhow, what do we mean by symbiotic microorganisms? Well, what you mean by that is basically an interaction between two or more species. So there are three types of symbiosis here, and the first one that they show here is mutualism. So you've got two organisms, both species will benefit upon that relationship. In this case, in our gut, we may have some microbes or bacteria that, because of what we eat, we are able to provide food for them. They, in exchange, may produce some products that we can use. That is mutualism. Commensalism, that is a fair amount of the microbes will fall in this category. And this is where one species benefits while the other is not affected at all. They're just hanging out. Okay, and many microbes operate by commensalism. We don't even know they're there. We don't really even care. But, they may be getting some benefit from being associated with us. Now, the third one is the one that gets a bad rap. That's parasitism. And that's where one species benefits while causing harm to the other organism. And, of course, this is the definition of a pathogen. Okay? The pathogen will benefit while we are harmed or our cells are harmed. So this is the picture, the overall picture. We've got our cells, which include tissues and organs, and includes tissues and organs. That's us on the left. See our little organs just hanging out there. Plus the microbes, the microbiomes, which includes your bacteria and yeast and viruses. So this is showing all those guys there. Okay? And their genes outnumber us. So finally, we are a composite of both, of our cells, tissues and organs, and the microbes that dwell within us and on us. That makes up our complete, total humanness. Okay? Now, how are we, or what capabilities do we have today in identifying these microbes? So in lab, what you're going to be doing is the tried, true, tested method of culturing. Back in the 1800s, this is the basic growing. You grow, test, and identify microbes using cultures. Very traditional method. But in addition to in the 21st century, we have additional methods due to the development of sequencing And at the gene level, at the protein level, at the metabolite level, and even at the messenger RNA level. So the first two boxes here, we have the 16S rRNA gene sequencing. So what they do is they break apart a little segment of a gene for rRNA, and it's the 16S, which is specific only to bacteria, rRNA gene segment. What they end up doing then is making many copies of it through the technology we've got today, and they sequence it, okay? They sequence the genes, nucleotides. And in this way, they are able to identify it. Another one that uses the genes is shotgun metagenomics, which is you're not using just a gene or a part of a gene. You are using all the genes or the whole genome of that microorganism. And in that way, by sequencing the whole genome, you are able to identify the organism. Next one up here in the pink box is studying the proteins that an organism may have. And that is usually from environmental sources. And you use some advanced technology for this. It really, you find this in research labs. You will not find it at ACC. And you won't even find it at the hospital. But many research organizations have the instruments, very expensive instruments to do this. And finally, well, not finally, but we've got in the green box metabiomics. So what this does is it kind of does a backway analysis in that it is analyzing the metabolites, the stuff, the products of these microorganisms. And by determining the products of these microorganisms, they can kind of work backwards in identifying the microbe. And then finally, you can profile a microbiome by seeing what type of messenger RNA it has. So these are all new technologies here, or newer technologies. And the important thing about this is, okay, once you identify, or once you've got a gene sequence, a protein sequence, a metabolite sequence, or even the messenger RNA sequence, what do you do with that information? And what that does is it goes into a database. So a sort of big computer library. It could be that database could be from a source out west. But anyhow, they've got all this information, and once the information you have determined is sent to this database, they will send back to you the identity of the microbe or microbes that are present. So this is really a lot of up-and-coming technology. And as a shout out for medical technology, which I've been in for about, 30-some years, this has come a long way from where I started in the 70s. And, they're always looking for a few good people. So if anyone's interested in this career, talk to me. There are a few schools that do offer it, but I can't say enough about technology and the medical technology field. So anyhow, parallel genomes. So what we've got here is in comparing the genome and the microbiome in us, some things are common and some things are quite different. We both have the same number of cells. So the total number of human cells in our body is about 40 million. The total number of a healthy microbiome is about 40 million. But if you look at the total number of genes, you'll see a difference. In our genome, we've got only 23,000 genes. In the microbiome, the total genes is 3 million. 3 million. And so what's happening? One thought is that, or one approach is that genes from the microbiome are actually controlling our genes or doing things that we are unable to do because of our limited number of genes. Anyhow, if you look at where we get our genome from, it's from parents, both parents. And actually, the microbiome is inherited from your mother, usually in the process of birth, also from the environment and other sources. We acquire that, inherit and acquire it. With the stability on our genome, it's pretty stable from individual to individual. So we've got 23 pair of chromosomes, pretty much the same for everyone. With a microbiome, there's a high variability between individuals. So I may have a certain amount of a bacteria in my gut in which a person standing next to me, well, maybe 6 feet away from me at this point, would have less of. So the number and the type of microbes may differ between individuals. Mutations in us cause disease. Mutations of our genome causes disease. With the microbiome, it actually is, disease can be caused by dysbiosis or an unbalance in the healthy microbes. And finally, I'm going to jump down to gene modulations. So in other words, can we modify the genes in our genome? There is some research in that, but it is risky. We don't know all of the results of that yet. Whereas in the human microbiome, you can modify it and even modify your microbiome. They're doing more research on this by targeting interventions. And I think one example of this would be with the fecal transplants. People with C. diff, an infection of the the intestine, the colon, they have very, very low amount of healthy microbes in that area. But now, and then in that case, the unhealthy or pathogens multiply and take hold. Well, what they've done now is they can transplant fecal specimens into patients and restore a healthy microbiome in the lower gut. So this is also very new and exciting research being done in that area. So if you want to read more about the human microbiome, it's in this research, in this article here. Again, some of the highlights is the human microbiome is an integral, important, very important part of the human body. Dysbiosis, when you are in unhealth or unbalance of your microbiome, diseases may result. Okay, and also certain strategies or certain environmental factors such as nutrition and antibiotics and supplementation with probiotics may provide new strategies with managing disease and prevention. So, one of the last of the two slides here is honing in on the effects of the gut microbiota. Okay, so we're back down in the gut because there are a lot of microbes that live down there. And in fact, when you look at feces, fecal samples, which I do in the lab, a healthy poop will be loaded with bacteria, loaded with it, because that's how the bacteria is excreted from our gut. Okay, bacteria live, they die, and they're excreted as poop. So anyhow, what they have found is actually there are some connections even with the microbiome in our gut. And they can affect the nervous system and the endocrine system. What happens is those microbes will release certain chemicals which can travel through the vagus nerve, spinal pathways, and they can affect our mood, our behavior. And also it's it's a two-way street, so the brain also can release some chemicals that will affect the endocrine system, such as ACTH and cortisol. Another responsibility or another job that the gut microbiota does is it maintains the immune system. Majority of our immune system lies right outside the gut, and microbiota, or the microbes, do maintain it, do kind of tweak it to keep it in check and to keep it healthy and responsive. If we do have a dysbiosis or an unbalance in the microbes in our gut, it has been linked to certain diseases. So what we have over here are some of the diseases we've got. It's been linked to arteriosclerosis, maybe diabetes, liver disease, irritable bowel disease, even obesity. Now the question is, which came first? The dysbiosis, the unbalance of microbes in the gut, or the disease which causes an unbalance in the microbes of the gut. So anyhow, I'm going to finish with this. We have two states of health and unhealth, and we're talking about the gut here. We have eubiosis versus dysbiosis. So eubiosis is the state of health, of homeostasis. And it really has been linked to certain diets that are rich in produce, such as vegetables and fruits, lack of fat meats. And what this does is they have found in the state of eubiosis that you've got the productions of certain components from these bacteria. They're short-chain fatty acids, which are used in our body for making other things. Also, they have found that there's an increase in eubiosis in microbial diversity. So you have many different types of microbes living in that gut, and also they are the good types of microbes. You will have also the decrease in certain types of microbes that can cause or be linked to disease, a leaky gut. Again, they do say that it depends upon, it is linked to Mediterranean or a good diet. Dysbiosis is unhealth, okay?